This is the what's it called? The cartridge, the Seahorse cartridge that we added calibrant to and left overnight to hydrate.
In this experiment, I am monitoring the glycolysis pathway in response to a panel of TAM receptor inhibitors and ligands and comparing them to untreated control samples.
So now we're just gonna prep the drugs that they provide in the glycolysis stress test kit. We have glucose, oligomycin, and 2-DG. So glucose is to increase the glycolysis of our cells, and it starts the glycolysis process. And then we add in oligomycin, which is an ATP synthase blocker, and so it forces the cells to keep doing glycolysis and shut everything else off. And once that's done, the third drug is 2-DG, which is a hexokinase blocker that is a rate-limiting enzyme in the glycolysis process. And so once this is given to the cells, they shut down their glycolytic pathway. Ta-da! After about 20 minutes of calibration in the machine spits out this plate which contains the calibrant, and once that's out, whatever plate that you have in the incubator with your cells, that's what you put in, and then it will start the whole process of injecting the drugs and measuring the glycolytic pathway. Is this 215, am I right? So this is the Agilent Seahorse machine. We can label where our cells are in the plate, which treatment we use for each, and once that's done, we can put in this entire thing, let it calibrate, and then come back with our cells. Calibrant plate. And with the cells, you load it. So once it adds glucose, we'll wait for around 24 minutes until it adds the second drug, and then it goes on like that.
An experiment like this can help us explore the role of TAM receptors in metabolic pathways and how these changes in metabolic pathways can affect cellular function, such as, in this case, the activation and antigen presentation of dendritic cells to cytotoxic T cells, which is an important step in immune cell action against cancer cells.
I'm really happy with this. And so this shows the first injection, the second, and the third.
Being a graduate student has taught me a lot about life as a scientist. Some experiments go as planned and many don't. This doesn't mean we're failing. It is an invitation to revisit the questions we're asking and tweak our experimental approaches accordingly. That, to me, is the beauty of an experiment, because the possible iterations are endless. Appreciating this uniquely creative opportunity to push the boundaries of science is important for success and happiness in this field.
And we're done. Thank you, cells.